Good morning, everyone. I'm talking about more specifically like tank conditions when it comes to optimizing the coral husbandry, how we're going to maintain the larval condition that we have in our tanks, hopefully for future conditions. The first step, our lab is the Coral Resilience Lab that is out of Kaneohe Bay in Oahu. So it's a little bit different. A lot of the focus at this presentation has been on Caribbean corals. This presentation, however, is on more Pacific. Our goal for our lab, at least, is to be able to get from the reef back to the lab and then back onto the reef with as much survival as possible throughout all of these steps. Over the last several decades, a lot of the big steps in coral restoration have been in asexual fragmentation, asexual propagation, fragmentation, and this provides a very easy, very repeatable source of material to place back on the reef. However, it can be seen to decrease genetic diversity within reefs, and it's also limited by material. Your broodstock can be reused, but you still need that initial material to use for that breedstock. Sexual propagation, however, can pull directly from the reef, provide an opportunity to maintain genetic diversity, as well as increase scalability. The more space you have, the more conditions you have, the more larvae you can rear, and hopefully the more larvae and settlement that you can place back on the reef. Now, our lab focuses on Montipora capitata. It is a hermaphroditic broadcast spawner. It releases both eggs and sperm in positively buoyant bundles. We will collect those spawn typically on the new moon, so it's a very repeatable, very followed schedule. They also are a vertical symbiont transfer species. The mother colony will seed her eggs with the symbionts from the colony, passing it on to the next generation. It is also an important colony for us because it is a dominant species in Kaneohe Bay, and it's also found throughout the Pacific Oceans, so it's very important and can be used throughout multiple islands. Now, our first step when we are practicing this is we'll go out to known reefs, known locations where the slick will kind of gather. So we'll send a team out with basic nets. We'll collect slick off the surface. Wild type, we don't know who's who, we don't know what's what. We just collect as much as we can and bring it back to the lab. Typically within the first hour, we try and get everything back to the lab into filtered seawater and prepared for fertilization, the breakup, and to get placed back into our conicals. Now, our conicals we oversee for about 5 days straight. I was about to try and do math, I really shouldn't. 5 days straight because during the process of fertilization and metamorphosis, it is possible for mortality to occur, and it is known that mortality can poison some of our conicals, so we have to be very careful in maintaining the chemistry within these conicals, because even losing just a few larvae can then breed a negative feedback loop and just kill the entire thing, so we have to be very careful. Once, however, you're at that end stage, you've got all of your larvae taken care of, the next step is trying to find the substrate to settle them on. Our lab uses aragonite plugs, and sort of the first part of the experimental process of these two papers that I'm working on, I'm talking about, are conditioning of these plugs. Conditioning and the generation of the biofilm, which was spoken about earlier, is very important because of these settlement cues. We want to know how long does it last for these settlement cues to sort of reach a peak state so that we can guarantee settlement. We did an experiment here. You can see in the graph to your left. On the y-axis is the number of recruits, x-axis is the amount of time that the plugs were left to condition. We did this over 1 week, over 4 weeks, and we also did another experiment over 10 weeks. And throughout all three of these experiments, we saw no significant difference between all three of our trials. So our lab has found the best practice of, just for efficiency's sake, going out, settling these guys, conditioning these plugs for a week, so you know you don't have to worry about it. Just throw them out, grab them, bring them back in. Now we've got our plugs conditioned, and we're ready to settle our larvae on them. Now, at what density do we want to settle these guys? We want to make sure that we settle as many as we can while keeping as many alive past that point that we can. So we did 3 treatments of 25 larvae per plug, 125 and 225 larvae per plug in our settlement bins that have about 80 plugs per bin. Now you can see here again, no real significant difference between our two higher conditions, the 125 and 225. And we also did another experiment to see over how much time we left them to settle. That's the 24 hours to 72 hours you can see on top of the rightmost graph in this experiment as well. And again, we didn't really see a big difference in settling them for an extended period of time. So our lab, we will settle as dense as we can, usually about 200 larvae per plug, and we will start pulling plugs at about 24 hours. Now, that's not to say that all the settlement happens within those 24 hours, but the majority will. Our lab's practice is to pull what does in that first day, leave whatever plugs are left and whatever larvae is left to see over the next coming days how many more can settle. Now our next step, we've got the larvae on plugs, and I'm going to start going into some of our survivorship data, is actually an explanation because you can see here circled in red aggregates and in blue individuals. We actually saw significant differences between some of our conditions in how these two different groups responded to those conditions. Now first here you can see just general survivorship over time. We did several experiments over the course of about 3 months. So you can see in blue on the y-axis we have survivorship, and on the bottom ag for aggregates and for individuals. The survivorship of aggregates was actually greater throughout all of our different treatments. However, throughout all of our treatments, here we have growth rate on the y-axis. Our individuals seem to grow faster. You will see, however, that the aggregates bar is slightly negative. When aggregates do come together, when we measure our growth rate, the aggregates are accustomed to partial mortality. Some of the polyps, as they group together, can die off. We no longer count those as a part of our growth, so we'll cut them out, and their surface area will get smaller. Now, briefly now, which I'll talk about later, during this experiment, which took place in 2019, Kaneohe Bay experienced a bleaching event, a natural bleaching event. Our system is fed through a filtered seawater system that's flow through. We pull water directly from Kaneohe Bay, pump through all of our tanks, and so all of our experiments actually experienced this bleaching event. We weren't planning on it, but we decided to take this data and see if we could find something interesting with our other treatments. So here you can see in aggregates versus individuals, the individuals more sharply responded to the bleaching event than the aggregates did. You will notice, however, the individuals seem to recover better. This is not as it seems. This drop in bleaching proportion in the individuals is actually due to mortality. The bleaching proportion is the number of alive corals that we have that showed bleaching once they died. We no longer counted them, which is why that number comes back down. Now we're getting into the actual treatments that we had for our experiments. This first is a flow rate. All of our tanks, again, are fed through our flow-through seawater system straight from the water from Kaneohe Bay. And we wanted to test if we could augment that flow and see if less flow, more flow provided a more beneficial environment for our corals. You can see on the y-axis mean survivorship, and in yellow we have our high flow tank, which had generally twice the amount of flow rate compared to the low flow. So you can see there's a significant difference between about 26% and about 10% survivorship between those two treatments. Higher flow is better. We want to keep as much water moving as we can, whether it's to provide food for the coral, moving sediment, or potentially disrupting the boundary layer between the coral. We're not exactly sure, but we believe probably somewhere in this realm. And this next flow we had was our light control. We wanted to see how much light we could restrict from the coral to prevent algae growth over our plugs. Our plugs are very accommodating towards carpeting algae that will overgrow the coral, so we wanted to see if we could restrict that as much as possible while maintaining survivorship. Now you can see in blue is our low light setting, so we had the most amount of shade over that, and the red is our highlight. And you can see a very interesting point on our little after a month time that there's actually a crossover where highlight had the opportunity for more algae growth. However, the low light seemed to restrict too much light from the coral to actually survive later on into this experiment. Throughout this experiment, the light was kept consistent, so the low light was low light throughout the entire time. That crossover point was really interesting to us. We wanted to see if maybe we could transition the light between times and see if we could maximize that growth rate continuing to go. You can see here in pink is the transition time, and in green is actually the highlight from the previous graph. You can see here. So we wanted to try in gray to see if maybe giving them even more light treated them even better, or maybe giving them slightly less, just to kind of vary it to see which treated them the best. And you can see between the highlight and even from the low light, the transition didn't have any significant difference. And overall, the original highlight treatment seemed to give general better growth rate. However, it was not statistically significant. Now, to compare if there was a trade-off between survivorship and growth, in aggregates, you can see a relatively straight line for our experiment, not so much. For the individuals, there is a slight decrease. However, it was not found to be statistically significant, though it is trending downward. Now, we're in the process of writing up this paper at the moment. Once we have all of our corals and everything taken care of, we want to see if we can reduce the amount of man hours that it takes to actually take care of them for the entire time. So here we have an experiment where we were using juvenile urchins to control algae overgrowth. And in this experiment, we left them completely alone. There was no human interaction between them. And we just wanted to see if the urchins could graze over the algae and make sure that the algae didn't overgrow the coral. You can see on the line, again, another negative growth rate. This is due to partial mortality. But you can see a significant increase in growth and survivorship between the urchins and the controls, where, again, there was no husbandry at all. So this is promising results. We've seen statistical significance, and we're looking forward to including more practices in hopefully more sort of naturalistic reef using other organisms to help maintain our coral larvae. Now, in the 2019 event, our corals went through 5.79 degree heating weeks of temperature change. And you can see here that our conditioning as well as our flow had an effect on their response to that bleaching event. So we're really looking forward to kind of going into more experimentation to see if anything else can affect or hopefully better prepare the larvae for such future events. I'd like to thank all the people in the lab. Josh Hancock and Sophia Ranka are the original two authors of the two papers I was using the data from today, as well as the other lab mates at the Coral Resilience Lab and the Paul G. Allen Foundation for funding for this experiment. Are there any questions?